Okay, guys, so I'm just going to kind of go through Avery's experiment just because when I went through it in lecture, I realized that I went through it and said, oh, you don't really need to know this. And then later on, I said, you need to know about Avery's experiment. So you need to understand Avery's experiment. So I'm going to give you a better explanation going through here. Now, before we go through Avery's explanation, I just need to recap Griffith's because Avery's experiment is based on some of the results that Griffith got. So this is just, the first slide here is just a recap of Griffith. So this is really what Griffith kind of showed, that S-type bacteria, okay, and S-type bacteria is the bacteria that can get you sick. Okay, I always remember the S-type is the one that makes you sick, or gives you pneumonia. So S-type bacteria, and again, the bacteria is a living cell, and all cells are made up of 4 components: protein, DNA, lipids, and carbs. So this bacteria is composed of these 4 macromolecules, and one of them is responsible for being able to pass on genetic information. Okay. Griffith really didn't know which one, but he basically proved that bacteria is capable of passing on genetic information. Okay. So in Griffith's experiment, basically what he did was he exposed one type of bacteria, S-type, that contains protein, DNA, lipids, and carbs, exposed it to another type of bacteria, which would also contain protein, DNA, lipids, and carbs. But when you combine these two together, these 2 types of bacteria, what he found was that something in the S-type, one of these macromolecules, whatever one it is, he had no idea which one it is, whatever one it is was able to pass on genetic information, be able to pass on code to R-type, and be able to convert R-type into S-type. Right? So let me just kind of... So what he was basically saying was that something in the S-type bacteria was able to transfer its information into the R-type and convert R-type into S-type. That's Griffith's experiment. Okay? So Avery's going to use this information. So let's see how Avery used this information. Go to a slide here. Okay? So this was what Avery did, was he basically said, okay, well, if I know that it's one of these 4 macromolecules, what if I start eliminating them one by one and seeing if it can still convert the R-type? So he said, okay, let's do this. If I destroy everything but protein, so he goes in and he says, okay, I'm going to destroy all the DNA, I'm going to destroy all the lipids, I'm going to destroy all the carbs. So the only thing that's left in this S-type is protein. Okay? And seeing now if it only contains protein, is it going to be able to convert R-type? And what they found was no, it was not. Okay? The bacteria remained R-type. So protein is not the molecule that's containing genetic information. Because if you only have protein, you can't carry that information into R-type. And it doesn't transform. Okay? So that's what he did the first time. Then he said, okay, let's do this again and say, okay, I'm going to now eliminate everything but, we're going to say lipids this time. Everything but lipids. So the only thing that's left now in this S-type is lipids. So he combines the S-type with the R-type and says, is lipids carrying that genetic information? Because if lipids were carrying that genetic information, it would convert R-type into S-type, but that did not happen. They still got R-type. So it's not lipids containing genetic information. So he said, okay, well, let's do this again. And let's destroy all the protein, let's destroy all the DNA, let's destroy all the lipids, and remain with carbs. I'm not sure why there's a G on the end of there, but again, you'll see lots of mistakes and grammar mistakes throughout this course. Okay, so we've got S-type, and now all that's left in this cell is carbs. So he said, okay, is carbs able to go in and transform R-type into S-type? And no, that did not happen. So he said, okay, well, let's do it this way then. Let's go through, we'll destroy all the protein, we'll destroy all the lipids, we'll destroy all the carbs, and then all that will be left is DNA. So is DNA able to go into R-type when they're mixed together and convert it? And yes, it was. So basically he confirmed that it's DNA able to carry on this information. Okay, now Hershey and Chase went one step further through their tagging, but this was kind of the results of Avery's experiment that I need you to know. Okay, thanks, and if you have any other questions, let me know.